Alright, so here we have our cell represented by a pool table. In the middle we've got the nucleus, the triangle being the nuclear envelope, and these lizards in the middle are the uncondensed DNA. Uncondensed.
First, the cell goes through interphase, where a gap phase occurs, followed by DNA synthesis, where the chromosomes replicate. This is then followed by another gap phase in preparation for meiosis. In meiosis 1, the diploid cell becomes two haploid cells through multiple steps. The first of these steps is prophase 1. In prophase 1, the nuclear envelope breaks down.
Boom. Broke down.
Where did it go?
It's gone.
And the DNA condenses.
Those were uncondensed DNA. These are condensed DNA called chromosomes. Notice how they're not all in the middle of the cell like they were earlier.
Next comes metaphase 1, where the homologous chromosomes line up along the metaphase plate. The homologous chromosomes split in anaphase 1. And two haploid cells are created in telophase in cytokinesis 1. Where the nuclear envelope slightly reforms and a cell wall forms between the two new nuclei.
These boxes represent the nuclear envelope forming around our chromosomes.
Next comes meiosis 2, where the sister chromatids separate to make a total of 4 gametes. The first step in prophase 2, where the nuclear envelope breaks down again. Next, the sister chromatids move to the center in metaphase 2. In anaphase 2, the sister chromatids split and move to opposite sides of the cell. Lastly, 4 gametes are created when nuclear envelopes are formed and the new cell walls are formed.
These pool sticks represent our cell walls, and as you can see here, we have 4 separate haploid gamete cells. Now, since both of the watermelons that we crossed in this problem here were identical, they're both heterozygous for both of their traits, this exact situation will happen again in another cell. And one of these 4 gametes will mate with one of those 4 gametes to make a whole new watermelon organism. The 4 possible haploid gamete cell genotypes shown in our demonstration of meiosis were arranged into a Punnett square. From this, we gathered the expected phenotypic and genotypic ratios of the F1 progeny.
From here, we conclude that an expected 9/16 of the fruit progeny will be bitter and spotted. 3/16 will be bitter with no spots, 3/16 sweet with spots, and as asked in the question, 1/16 of the fruit will be sweet without spots. Let's see how this plays out in real life.
Now that's a sweet watermelon.
Watermelon number 2.
Alright, you ready? God! Oh, that's bitter. Yeah.
Oh yeah, that is definitely a dominant tasting. Oh.
No watermelons were harmed in the making of this video. Just kidding. We destroyed them.